Hello, everyone. Welcome to today's live webcast, Next Generation HPMC Capsules, Bioequivalence and Functional Performance. We're pleased to bring you this educational web-based event presented by Pharmaceutical Technology and sponsored by Capsugel, a global leader in delivering high-quality, innovative dosage forms and solutions to healthcare companies around the world. Their customers turn to them for the science and engineering expertise they need to bring better medicines and nutritionals to market. As the world's leading provider of empty, two-piece hard capsules and an innovator of drug delivery systems, they continue to build on a decades-long track record of quality, reliability, innovation, and value creation. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can after the presentations. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of the presentation window. Now I'd like to introduce today's speakers. Joining me today are Matt Richardson, PhD, and Michael Morgan, PhD. Matt Richardson is the Manager of Pharmaceutical Business Development at Capsugel. A graduate of Wofford College, he holds a doctorate from Wake Forest University and a postdoctoral appointment at Virginia Tech. Before joining Capsugel, he was with Schweitzer Hall and Irex Pharmaceuticals. Since joining Capsugel, he has worked with many pharmaceutical and nutraceutical customers to understand the formulation aspects of their products with two-piece hard capsules. Matt's been an invited speaker at numerous seminars and webinars on this topic. Michael Morgan is a senior principal scientist at Bend Research, a division of Capsugel dosage form solutions, where he's worked since 2003. He has more than 15 years of experience in the development and evaluation of novel polymeric materials, including pharmaceutical formulations for oral and non-oral delivery, particularly for low solubility molecules. He received a doctorate in physical chemistry from the University of California at Berkeley. He is co-inventor on 14 patents and patent applications and co-authored 21 papers and book chapters. Thank you both for being here today. And Matt, please go ahead and get us started.
Thank you, Rita. And thank you to everyone that's joined our webinar today. In the webinar, we want to cover what makes next-generation HPMC capsules so different from either existing HPMC capsules or also hard gelatin capsules as well. We're going to do a little comparison as far as that goes. In the webinar, we want to cover that two-piece capsules made from HPMC have been used as an alternative to gelatin. And traditionally, that's been when something has challenged a gelatin, something like an incompatible active or hygroscopic or moisture-sensitive active. New HPMC polymers have eliminated the use of secondary gelling agents and instead use a thermo-gelation to form that film. And that results in a performance that's much, much closer to what you'll find with a traditional gelatin. So this webinar is going to address the characteristic of these next-generation HPMC capsules. And we're also excited to talk a little bit about some new in vivo performance data, and that's going to show the comparison of HGC, hard gelatin capsules, versus HPMC as well. HPMC is not a new material to pharmaceuticals in and of itself. It has been used and well vetted in pharmaceutical formulations for many years. Similarly, capsules made of HPMC, they're not novel either. They have been part of the pharmaceutical industry for several years. Recent advances in the development and manufacture of HPMC capsules, however, That's led to a huge expansion of their use, well past what we have seen in previous years. An increasing percentage of new compounds are being evaluated in HPMC capsules at the onset of formulation studies, either as primary, the capsule of choice, or at least in simultaneous studies with hard gelatin capsules. This is a true change in the dynamic of capsule formulations that in previously HPMC capsules were really only considered as a last resort or a fail-safe formulation option. So I'd like to spend a little time introducing why that shift is occurring. First, let's look at a bit of background information on gelatin. Gelatin is produced from the hydrolysis of collagen derived from the bones and skin of both beef and pork. Gelatin is ultimately a chain of amino acids. It needs no additive to gel on its own, what is termed self-gelling, which occurs at a particular concentration when the temperature of the solution reaches below 40 degrees Celsius. As it comes out of solution, it forms a three-dimensional chain network, as seen in this structure. After drying, the capsule has about 12 to 15% water content. The water content is important for gelatin in order for it to stay nice and flexible. When the water content goes out of that range significantly, it has an effect on the performance of gelatin.
Matt, this is Rita. Matt, this is Rita. Could you speak up a little more?
Sure.
If you just speak a little louder, that'd be thanks.
Okay. Thank you. HPMC capsules, in contrast, is a cellulose-based material derived from plant sources, usually wood pulp. That cellulose raw material is extracted from the plant material, and then its existing hydroxyl groups are partially substituted to afford the final HPMC raw material by the process shown in the lower right of this slide. Depending on the degree of substitution and viscosity of the products, HPMC can then be further separated and classified into different grades associated with both chain length of the polymer and also viscosity. HPMC capsules have been marketed for more than 10 years. Those first HPMC capsules were made under similar process conditions as hard gelatin capsules, utilizing cooled stainless steel pens, which are dipped into a hot solution of HPMC. The solubility of HPMC, however, is lower in hot water than cool water. So to help stabilize the polymer in solution, gelling systems comprised of both a gelling agent and a gelling promoter must be added to the mixture. As customers globally tested that first generation HPMC capsule, they experienced some variability in physical attributes, but most notably in the dissolution of their encapsulated formulations. As a result, they relayed both their concerns and interest in capsules which could demonstrate less variability. Upon further investigation, we were able to show that the variability could be attributed to the addition of a gelling agent and gel promoter. It was clear that an alternative process was needed in which gelling systems were not used at all. This led to research on the manufacture of HPMC capsules without gelling systems involved, taking advantage of the solubility of HPMC in cool water, which allowed for a more uniform, concentrated solution of HPMC. Heated stainless steel pens are dipped into the HPMC solution, allowing for the HPMC to deposit as a solid film on the stainless steel. This required a complete reversal of the traditional hard capsule manufacturing process and the equipment to produce this second-generation HPMC capsule. We at Capsugel use the trade name Vcaps Plus for these second-generation capsules, and I'll use that name often throughout the presentation to refer to capsules which are made by the thermo-gelation process and do not contain any gelling systems. After drying, capsules are removed from the stainless steel pens, assembled, and then finalized much like any other two-piece hard capsule would be, regardless of the polymer. Whether made by that first generation process in which gelling systems are required, or the second generation, the Vcaps Plus capsule process, which completely avoids the addition of gelling system, HPMC capsules have properties that are quite unique relative to themselves and to gelatin as well. In this section, I want to cover the physicochemical differences that can be expected between the polymers as a guide to help formulators on which material is most suitable for their particular formulation. With respect to the dimensions of the capsule of the different polymer types, both HPMC capsules are identical to the size and shape of gelatin capsule, and there's no difference, therefore, seen in this regard for either formulation or for manufacturing. Capsule weight specifications are similar to hard gelatin capsules as well, with target weights matching quite closely. Technical reference files are available for each polymer for a review of both of these elements. A vast array of color and imprint choices are available through both the gelatin and the Vcaps Plus capsule. Colors across the spectrum have already been both developed and validated for use in commercial manufacture. Water content, as we can see in this particular slide, however, is the first most notable difference between the physical nature of the HPMC and the gelatin capsule. In the graph on the left, you can see a moisture content for hard gelatin capsules. And what you'll see is it's ideal at 12 to 16%. That represents the safe storage window of about 35 to 65% relative humidity for a gelatin capsule. This is a fairly fixed window for a gelatin capsule optimal performance. Should the moisture level in the capsule drop too low, concerns of brittleness are heightened. On the other end of the bookcase, as we call it, capsules can gain too much moisture and then begin to lose their shape. The impact of this is evident in manufacturing, showing up as negatively impacting encapsulation speed. As long as the hard gelatin capsule is kept within the recommended storage conditions, it is a very robust capsule. With HPMC capsules, however, the different polymer lends to a different result. In the graph on the right, you can see the two major differences. First is that the ideal moisture range is 2 to 9% for an HPMC, allowing for a wider operating range according to the relative humidity. Secondly, the average moisture content for HPMC capsules is about 5 to 6% water, compared to that of gelatin at 14 to 15% water. No disparity in the moisture levels is noted when the absorption and desorption isotherms for both HPMC and gelatin are examined as well. So here you have the gelatin in the blue, and you have the Vcaps Plus, the second generation HPMC, in the red. And this corroborates the overall moisture data demonstrated in the previous slide and represents to the formulator an opportunity. When either moisture-sensitive or hygroscopic actives are being formulated, this differential in water content would make a very significant difference. We tested the viability of HPMC capsules giving additional protection and stability with trials using the very moisture-sensitive compound, acetylsalicylic acid. In this experiment, acetylsalicylic acid formulation was placed in both HPMC and gelatin hard capsules on two stability platforms, 25 degrees and 60% relative humidity, and also 40 degrees Celsius at 75% relative humidity. The capsules were placed in inductively sealed HDPE bottles so that environmental concerns were negligible, and the amount of degradation was then measured over time. In both cases, it can be seen that the amount of degradation is higher in the gelatin capsule as compared to the HPMC capsule shell. After 18 months at the 25 degrees 60% relative humidity stability, there's approximately 8% degradation found in the formulation contained in the hard gelatin capsule as opposed to the 2% in the HPMC hard capsule. Similarly, at accelerated stability, the same effect can be seen at the 6-month time. Approximately 5% of degradation in the formulation at the hard gelatin capsule as opposed to just over 1% found in the HPMC capsule. As the capsules were inductively sealed, we can effectively rule out any contribution from the environment. so the moisture found in the capsule itself is responsible for any product degradation. As we saw in the previous slide, HPMC has about 6% median water compared to that of gelatin's 14%, and this would explain the differential in degradation due to moisture. I showed earlier that when the moisture level begins to drop too low for hard gelatin capsules, the shell begins to lose its pliability and would then be more prone to brittleness and then possibly breakage. In this slide, I want to show how hard gelatin and HPMC capsules compare to each other with that regard. This can be done utilizing a tube test device, and that's pictured on the upper left of this slide. The device drops a 100-gram weight, a distance of 8 centimeters, onto the capsule, and the resultant capsule is examined for signs of cracking or shattering. A sample of 50 capsules, each of HPMC and gelatin, was equilibrated at various shown relative humidities and then subjected to the tube test evaluation. The graph on the right side shows the percentage of broken or damaged capsules at the various equilibrated relative humidity levels. The trend is quite visible with an increasing percentage of gelatin capsules breaking as the relative humidity levels drop, and that corresponds to a lower amount of moisture in the gelatin shell. HPMC, however, does not show the same level of brittleness at the lower humidity levels. Whereas gelatin utilizes the water as a plasticizer to remain flexible, HPMC does not, and as a result, maintains its robust nature at lower moisture levels. This allows for storage in drier conditions, as well as has significant implications when desiccants are used to protect formulations as well. Another characteristic that shows an interesting difference between the two HPMC polymers is weight variation. Gelatin, seen on the right side of it's the rightmost column of that table, it has the lowest amount of variability in weight. Recall that gelatin has been manufactured for decades prior to the start of production of HPMC capsules. That's allowed hard gelatin capsules to set the bar from a manufacturing perspective. Vcaps Plus capsules, manufactured through the thermo-gelation, and that's designated as HPMC Capsule Shell 2, also shows a better weight consistency than the HPMC capsule, which does contain a gelling system. That's designated as the HPMC Shell 1. In this review, conducted by Koo and others, the Vcaps Plus HPMC capsule showed a 3.29% relative standard deviation compared with the first generation capsule containing a gelling system which showed an almost twice weight distribution at 6.45 relative standard deviation. This would suggest that capsule to capsule and batch to batch variability would be lower using capsules, using HPMC capsules that do not contain gelling systems. Scanning electron microscopy of shell surfaces across the three polymer types are presented in this slide. Vcaps Plus, again shown in the slide as HPMC Shell 2, has a clean edge and very smooth surfaces compared to both the hard gelatin capsule and the first generation HPMC capsule. The maximum gap between the body and the cap, as well as the measurement of the shell thickness at the closure, is summarized in the photo. The microscopy shows hard gelatin capsules have the thickest shell, followed by HPMC Shell 2, the Vcaps Plus, and HPMC Shell 1, the first generation HPMC capsule containing a gelling agent. Microscopy also shows that while the shell thickness variations by relative standard deviation are similar, hypermellow Shell 1 has a large gap between the body and the cap, twice as much as the gap for that of the gelatin capsule. HPMC Shell number 2, the Vcaps Plus, has a slightly larger gap than the gelatin, but is still much smaller than what is found in the hypermellow Shell 1. The reference paper describes how improvement in the powder leakage quality attribute can be expected from the HPMC without the gelling system. That's shell number 2. And when trialed against the other shells, no powder leakage was observed when HPMC shell number 2 was used, while leakage was evident with the first generation shell, the HPMC shell number 1. The evenness and smoothness of that HPMC film also contribute significantly to the higher quality performance of the HPMC shell number 2 in manufacturing conditions and environments as well. Another notable difference is the ability for the HPMC capsules to better tolerate heat deviation or hot storage conditions such as ICH Zone 3 and Zone 4 stability regions. Recommended storage conditions for gelatin capsules would be placed in the 15 to 25 degrees Celsius range, and deviations are certainly permitted with accompanied stability data. As Zone 3 and Zone 4 call for 30-degree heat storage, this is somewhat out of range and therefore more prone to instances of either cross-linking or heat damage as a result. HPMC capsules actually show heat resistance well past that of gelatin. And you can see in the graph there's no change in the HPMC, the Vcaps Plus capsule, even up as high as 60, 70, and 80 degrees. Now that we've taken a look at some of the physical attributes of the capsule polymer, I wanted to focus in on the disintegration and dissolution characteristics of the materials. In this aspect, we see the greatest impact of the gelling systems on the performance of hard capsules, and probably the largest amount of questions from customers as well. Understanding the impact of gelling systems on the first generation HPMC capsule on disintegration and dissolution drove the production of the manufacture of the second generation HPMC capsule. And so the goal was to have an alternative polymer capsule with great disintegration and dissolution performance with little to no variability. In the inset of the graph, a smaller chart showing the disintegration rate of Vcaps Plus shows that it is very similar but slightly higher compared to gelatin. This effect is more pronounced when one compares the dissolution rates of caffeine in both gelatin and Vcaps Plus thermo-gelation HPMC capsules. Here, it's noted that gelatin quickly releases the contents, allowing for dissolution to manifest rapidly, while Vcaps Plus does not appear to allow dissolution until approximately 5 minutes into dissolution testing. It turns out to be a phenomenon occurring in all commercially available HPMC capsules, either first or second generation. Recall that HPMC capsules have less water than gelatin capsules, and as a result, HPMC needs a hydration period in order to pull water into the matrix and allow disintegration to begin. Gelatin already contains 12 to 16% water, well, it doesn't need the same hydration period, and that accounts for the difference seen. It is important to note a couple of things here. First, the percentage of caffeine in solution is equivalent at about 15 minutes, and that's indicative of the fast opening of the second generation of HPMC capsule despite its initial lag time. Secondly, if the dissolution was not compared to gelatin at all, which really shouldn't be, given that it's a completely different chemical polymer, the Vcaps Plus capsule would still be considered to have a fast disintegration time. Gelatin has been the benchmark, however, and so it's interesting to see the comparison between the two. This graph shows dissolution variability that shows up in HPMC capsules that contain gelling systems in them. HPMC capsules using carrageenan and potassium salts as the gelling system were tested. A variety of dissolution media were tested to produce the data shown in the graph. As the differences in the media are investigated further, the implications of the variability begin to show up more fully. Consider first the dissolution curves comparing pH 1.2 by USP and pH 6.8 USP. At first glance, it appears that a difference in pH is responsible for the high amount of variability between the two curves. But notice this pH 6.8 buffer system made according to the JP2, the Japanese 2 protocol, gives a very similar dissolution to that of the pH 1.2 by USP. Its pH is not a significant contributor to a gelling system that uses carrageenan. Rather, what is different between the pH 6.8 buffers made by the USP and JP2 protocols is the ionic activity between the two systems. The JP2 protocol was created to have a lesser ionic strength because of the contribution to variability and dissolution that arises from secondary gelling systems found in HPMC, the first generation HPMC capsules. This is further substantiated when you start taking a look at the dissolution curves of pH 1.2 by USP versus those of pH 1.2 in which potassium chloride has been added at 2 and 9 gram quantities to increase the ionic strength of the buffers, which in turn show an increasing amount of variability in dissolution. So if variability is noted in vitro with ionic media, then an in vivo concern should be suspect as well. Diets like the American breakfast, in which fruit juice, or vitamin consumption is part of, would naturally lend to variability in release rates for HPMC capsules that contain a gelling agent. Simulated milk fluid is routinely used to compare the ionic strength of the fed state against that of the fasted protocol by the pH 1.2. And you can see that the variability is there between those graphs as well. Now, contrast the data in the previous slide with the data shown in this slide. Using the same protocols and media previously used, but changing to a Vcaps Plus capsule, which contains no gelling system, producing dissolution data with far less variability than previously seen. And I'll just go back one to let you see the disparity in the two slides once more. Okay? The massive differences that were shown previously, according to ionic strength and the food effect, have resolved into very similar data when no gelling system is used. If the dissolution graph is studied, no particular trend is evident based on the dissolution media when a Vcaps Plus capsule is used, suggesting that the removal of the gelling system is paramount in reducing variability. This is not to say there's no variability displayed, but however, the variability shown is neither significant nor attributed to a particular interaction. It is believed that the initial rupture time of the capsule is slightly different, then some of the change in the relative standard deviation could be noticed as seen here. Currently, investigation in the moisture content of the capsules is being investigated to produce an even smaller relative standard deviation. In a study presented in the International Journal of Pharmaceutics, Hu and others also showed a dose escalation experiment in humans to investigate the performance of Vcaps Plus in vivo. The study demonstrated both that at the 5-minute delay seen in vitro had no bearing in vivo, and more importantly, that the capsule opened consistently across the dose escalation range with a median Tmax observed at each case in 1 hour. This is indicative that the shell disintegrates quickly, allowing for the subsequent dissolution and absorption of the immediate release compound tested. The results of that study confirmed that the capsules had an excellent performance in vivo and further confirmed the viability of Vcaps Plus HPMC capsule made by thermo-gelation. So to further study the performance with different types of compounds and also to understand its in vivo performance as compared to the gelatin benchmark, we also wanted to take a look at its performance in vivo and want to share some of the results of a study that was recently performed. The study design was an open-label, single-dose, randomized, two-way crossover that was done with 24 healthy male subjects. Excedrin extra-strength caplets were chosen as the dosing material, as the caplets contained 3 different markers that could be tracked in vivo. Interestingly enough, the 3 markers represent 3 different biopharmaceutical classifications by the BCS system, and that would further add to the study. The protocol was having patients take 2 Excedrin extra-strength caplets, which had been encapsulated in either hard gelatin capsule or the Vcap Plus HPMC capsule that contained no gelling agent. The compounds were tested in vitro by the monograph for the acetaminophen, aspirin, and caffeine tablet, which called for the use of the USP dissolution apparatus too. And you can see the pharmacokinetic parameters that were measured during the in vivo study listed on this one as well. So first we'll take a look at the results for the acetaminophen PK parameters. Those are shown here. At the top is the graph of the mean plasma concentration of acetaminophen over time. The plot for the hard gelatin capsule is shown in red, the plot for the Vcap Plus HPMC capsule is shown in blue, and the results are near identical for the mean plasma concentration levels. The statistical analysis is shown in the table below the graph. Cmax and area under the curve values match up quite nicely for acetaminophen as well, and give a high confidence interval showing the equivalency of the encapsulated forms of acetaminophen between hard gelatin and the thermo-gelation HPMC capsule containing no gelling system. The results for the acetylsalicylic acid PK parameters are shown in this slide. Again, at the top is the graph of the mean plasma concentration of aspirin over time. The plot for hard gelatin capsules is shown in red, the Vcaps Plus HPMC capsule shown in blue. For this compound, again, a separate classification by the BCS system, the results show a near identical match for the mean plasma concentration levels. Looking at the statistical analysis in the table below, we see again a good match between the Cmax and area under the curve values for the aspirin. The intrasubject variability for the Cmax is a little high, but that's not atypical for aspirin. If you look at the area under the curve values, you see quite high confidence intervals. Collectively then, the equivalency of the encapsulated forms of acetylsalicylic acid, whether in hard gelatin or Vcaps Plus HPMC, is again demonstrated. And lastly, we'll take a look at the results for the caffeine PK parameters, and shown in this slide. In the graph of the mean plasma concentration, caffeine over time, you get a very similar data trend when comparing the product encapsulated in hard gelatin capsule, again, that's the one in red, versus that of the VCP, the Vcaps Plus HPMC capsule shown in blue. The statistical analysis in the table below, again, show good data similar to the previous two, matching values in the Cmax and area under the curve with good confidence intervals. Caffeine being the third molecule and the third different type compound in the BCS classification to show in vivo equivalency between encapsulated forms in hard gelatin and HPMC without a gelling agent, we can then conclude that there's no significant in vivo difference that comes from using an HPMC capsule that contains no gelling system. Only made of HPMC and water, interaction between the capsule and the dissolution environment, whether in vitro or in vivo, is negligible. So in summary for this part, HPMC capsules have been shown to broaden the space for two-piece capsule applications in both pharmaceutical product development and also manufacturing. Characteristics and performance of HPMC capsules differ because of their manufacturing process, some being manufactured through a thermo-gelation process very closely reaching gelatin-like quality and also shown in vivo performance. In vitro dissolution and in vivo bioequivalency studies demonstrate that the Vcaps Plus product is a good candidate for new APIs currently in development in gelatin, as well as potential switches for products registered in gelatin to the more robust HPMC capsule. And with this, I'm going to conclude my part, and I'd like to turn it over to Mike Morgan and let him talk to you a little bit about some of the in vitro modeling and early research that has been going on at Bend. Mike?
Thank you, Matt. I'd like to, as Matt said, talk a little bit about some modeling we've done around the in vivo study that Matt just described, and then talk a little bit about applications of using HPMC-based capsules as functional excipients. So again, here's the in vitro dissolution data that we saw for the 3 actives that were in Excedrin. As we've seen, the profiles for the 2 capsules are pretty similar, with just a few minutes delay for the caplet encapsulated in HPMC versus that in gelatin. We went ahead and did some fairly straightforward pharmacokinetic modeling of one of the 3 compounds, acetaminophen, to show that the in vivo profiles obtained were consistent with expectations, but also to show the sensitivity of the PK profiles to the drug release profiles from the capsules. And in this modeling, we varied 2 things. First, we varied the lag time for release of drug from the capsule while keeping the dissolution rate constant, which is essentially what was observed in vitro. We then varied the dissolution rate while keeping the lag time constant. And although we didn't see big dissolution rate differences between HPMC and gelatin for these soluble compounds that are in Excedrin, we wanted to show the sensitivity to the dissolution profiles. So this slide shows the effect of dissolution lag time. The actual in vitro dissolution curves from HPMC and gelatin capsules are shown in the upper left, which are fit to a Weibull function. The upper right curve then shows the resulting simulated, or upper right plot shows the resulting simulated PK curves for various lag times ranging from a few minutes to 2 hours. The bottom plot shows the effect of dissolution lag time on Cmax, AUC, and Tmax. And as expected, the Cmax and AUC don't change appreciably, whereas the Tmax gets pushed out to longer times with increasing lag times, as one would expect. Now on this slide, we show the results for the constant lag time with varying dissolution rate constant. Again, we didn't see significant experimental differences in the rate for Excedrin from the 2 capsule types, but this modeling was done to demonstrate the sensitivity to rate that one might expect. So we show curves with various rates on the upper left plot, ranging from a few minutes to 4 hours. On the top right are the simulated PK profiles. And then in the lower right plot, you can see that the AUC is not very sensitive to dissolution rate for highly soluble, well-absorbed drugs like acetaminophen. The Cmax is more affected, but only drops about 10% for a dissolution rate as slow as 90% release in 75 minutes. So we've seen that the soluble compounds that are well absorbed in Excedrin have PK profiles for HPMC-based Vcaps Plus and gelatin capsules that are expected to be similar. One might ask if there are applications for which the 2 capsule materials might perform differently, and indeed there are some such situations. One mechanism that can lead to differences in performance is favorable HPMC drug interactions that lead to reduced crystallization rate of drugs in the GI tract for poorly soluble drugs when using HPMC-based capsules. And this effect can be important when there are supersaturated drugs in the intestine, which can result from dosing either a high-energy salt form, an amorphous form, or a weakly basic API, where there's a concentration of dissolved API in the gastric that leads to a significant supersaturation upon transit of the drug to the small intestine. In these cases, we found that HPMC-based capsules can help to maintain that supersaturation by inhibiting crystallization of the drug. Okay, so today I'll talk a little more in detail about one of these applications, that of weakly basic drugs. In the next few slides, we show examples of dosing several weakly basic drugs into a simulated gastric environment and then looking for sustainment as that media is transferred to a high pH simulated intestinal fluid where they convert back to lower solubility free base forms. In these studies, we compare the performance in Vcaps Plus versus gelatin. And of course, we would expect the sustainment of the drug to depend on the drug concentration relative to the gastric and intestinal solubilities of the drug, as well as on the tendency of the API to crystallize, and on the type of capsule used. And as you'll see in the following slides, for all 4 of the drugs we investigated, we saw improvement of sustainment using the HPMC-based capsules relative to gelatin. Next slide, please. I don't know if somebody can advance this. Okay. Sorry, my screen went down.
Oh yeah, Mike. This is Rita. We're on the weekly basic drugs slide is showing right now.
Yeah, I see it. Sorry, I had some technical difficulties here with my screen. So here are listed the 4 compounds we used. You can see that they're all relatively soluble in the ionized state in the low pH gastric environment, but have lower solubility at neutral intestinal pH, which can lead to a highly supersaturated state of the drug in the intestinal environment in vivo. Next slide, please. Here we show the results for dipyridamole and erlotinib. The green curve in these plots is from the API and gelatin capsules, and the blue curve is API and HPMC-based Vcaps Plus. And then on the x-axis, from 0 to 30 minutes represents the time spent in the simulated gastric fluid, and then there's a transfer at 30 minutes to the simulated intestinal fluid. And there's a discontinuity at 30 minutes because there's a 2-fold dilution upon transfer from the simulated gastric to simulated intestinal fluid. So the initial concentration drop at 30 minutes is from that dilution. And you can see that the API sustains better in the simulated intestinal fluid using the HPMC-based Vcaps Plus versus the gelatin capsules for both of these compounds. Okay, this slide shows the second 2 compounds, ketoconazole and gefitinib. And again, you can see with the blue curves from the Vcaps Plus encapsulated drug sustains better in the simulated intestinal fluid than does the gelatin-based capsules. So we also studied the drug sustainment as a function of drug concentration, which can have an effect on the crystallization precipitation kinetics. And shown here is data for erlotinib, where in this case we have added pre-dissolved drug at various concentrations to the solution, either with or without pre-dissolved HPMC. So the HPMC is pre-dissolved at 0.1 mg per mil, which is a relevant concentration for a dissolved capsule. And you can see that the sustainment depends on the drug concentration and is better with the pre-dissolved HPMC, which is shown on the left plot, than the experiment without HPMC, which is shown on the right plot. So a fair question would be whether these differences in sustainment observed in vitro are manifested in vivo. And this slide shows an example from the literature where the mesylate salt of dabrafenib was dosed clinically in both an HPMC and a gelatin capsule. And the top plot shows that the AUC was significantly higher for the drug encapsulated in the HPMC capsule. The authors performed in vitro dissolution tests in simulated intestinal fluid, shown in the bottom of this bottom plot, that showed that the sustained drug concentration was higher for the HPMC capsule, likely due to the better crystallization inhibition of HPMC. So again, the degree to which crystallization inhibition affects in vivo performance will depend on the particular application, but HPMC has the potential to play a role as a functional excipient to inhibit crystallization and help sustain the concentration of supersaturated drugs in the GI tract. So this potential for a functional capsule shell to sustain free drug levels is of interest because a large fraction of drug molecules in development are either weakly basic or have poor solubility and are therefore likely to be formulated as a high-energy supersaturating drug form. So we've looked at our recent portfolio of molecules formulated here at Bend and determined that of the last 500 or so molecules we've seen, approximately 40% of them are weakly basic. That is, having a basic pKa between, say, 2 and 7. And almost all of those compounds are poorly water soluble, as shown in the plot here, which displays the solubility in FASF versus the basic pKa. And the orange box is shown around the compounds that have a physiologically relevant basic pKa of somewhere between 2 and 7. So there are quite a few compounds that are potentially can benefit from HPMC-based capsules, and the opportunity space is larger even than what I just showed, because as I said, many non-basic drug compounds have low solubility and are therefore formulated as a high-energy form, such as an amorphous dispersion or a high-energy salt. And these forms can supersaturate in the GI tract and potentially benefit from crystallization inhibition. So shown here is the well-known developability classification system, with some estimates indicating that a majority of the drug compounds in development, maybe as high as 80% in some portfolios, have poor water solubility. And these are the ones that could potentially benefit from formulation as high-energy forms, and that may also benefit from crystallization inhibition from polymeric materials such as HPMC in the capsule shell. So just to summarize the latter part of the presentation, gelatin and Vcaps Plus capsules are likely to give similar performance for a broad range of applications where the API is relatively soluble and rapidly absorbed. But there are certain applications, such as for weakly basic crystalline APIs or for amorphous forms or low solubility APIs, where Vcaps Plus capsules may improve solubilization and precipitation inhibition. And this is one of the areas that Capsugel continues to actively study and to understand better the applications there. And Capsugel is engaged in research to understand and reliably predict from in vitro data the relative in vivo performance of active ingredients and formulations encapsulated in the 2 capsule types for various applications. And of course, we're glad to engage with clients on specific applications that they may have in mind. So with that, I'll end and turn it back to Rita.
Well, thank you, Matt and Mike, for those informative presentations. We are now ready for the Q&A portion of the webcast. Audience members, you can type your questions in the widget located at the bottom of your presentation window. And Matt, we'll start out with you. What is the recommended storage condition for HPMC capsules in terms of percent RH?
So what we'll say, the packaging will probably tell you is similar but you actually have a wider window. You could do packaging, storage anywhere from the 15% up to, I would say, a good 65 to 75%. I wouldn't spend a lot of time at 75%, so I recommended the most favorable window is still going to be in a similar 30 to 65, but you could effectively get away with anything from a 15 to a 70% range. So that opens it up quite a bit.
Okay, thank you. Here's another question for you, Matt. Is there any comparison for moisture vapor transmission rate through a film made of HGC versus the HPMC caps?
There is, and it's actually very similar. So the moisture vapor transmission rate is pretty close between a gelatin and the HPMC as well. So its effect in storage at a different relative humidity, it's going to pick up or it's going to lose moisture pretty much the same rate between either a gelatin or an HPMC. The difference is that the gelatin, as we said in the presentation, has pretty well-defined window of operability that you want to stay in, and the HPMC has a larger one. And while I don't have the moisture vapor transmission rates on the top of my head that I could, if you contact me, I'm happy to send you those.
All right, thank you. Mike, we have one for you. What formulation or other application parameters determine the degree to which HPMC-based capsules improve sustainment of dissolved drug levels?
Yeah, so as we were saying, the drug compounds for which that might come into play are low solubility drugs, and particularly those that can supersaturate in the gastric, and those include weakly basic compounds, whether dosed as a high energy form or as the crystalline free base, as well as high energy salts and amorphous forms. So it largely depends on the type of drug compound. But those compounds that are low solubility and can supersaturate, and especially those that have a high tendency to crystallize, such as compounds with high melting points, are the ones that are probably the best place to look for improvement from HPMC-based capsules.
Okay, thank you. Matt, can the same technology be used for soft gelatin capsules?
If we're talking about the same technology as far as the first versus the second generation HPMC, I'm actually not aware of that being applied to soft gel technology today. And the reason is all around the film forming properties of the HPMC, and especially the thermo-gelation HPMC. So a soft gel made by the HPMC probably wouldn't tolerate the thermo-gelation process that the hard gelatin can.
Okay, thank you. Mike, how typical is a weak base molecule?
In our experience here at Dosage Form Solutions, it's very typical. As I said, looking at the last several hundred compounds that have come through our facility, something like 40% of them are weakly basic low solubility compounds, mostly BCS class 2. So in our experience, it's a very common API type.
Okay, thank you. Well, let's go back to Matt. Are there any mechanical or capsule filling machine differences between HPMC and gelatin?
No, there really aren't. So we did do some due diligence around the filling machine. So our technical service group actually did a review of 32 different filling operations, and probably now it's a much larger number, across really a variety, either intermittent motion and semi-automatic machines as well. And material filled in Vcaps Plus gave performance, I would say average yields are right in that high 99% rate. I believe the range was anywhere from 99.5 up to 100%. So in essence, you will not experience any slowdown, and you won't have any, or we haven't found any significant differences in manufacturing using an HPMC thermo-gelation versus hard gelatin. I will say that there's probably some tweaks that are going to be involved, and you'll have to take a look at some of those recommendations from technical services. HPMC is a softer material, so some of the settings on the machine may say closing force and things like that. Those may be adjusted, but it typically doesn't require any capital expenditure, just machine settings that are already there. We've got a technical service team that are happy to make those recommendations whenever.
Okay, thank you. Mike, for weak bases, step dissolution, was 100% dissolved at 30 minutes when transferred to FSIF? And if yes, what happens if incompletely dissolved material is transferred to a higher pH?
Yeah, so in the examples we showed an example of 4 model compounds. In some cases, 100% was dissolved, and in some cases not. And of course, the precipitation kinetics that happen upon transfer will depend upon the relative solubility in the simulated gastric, as well as the solubility in the intestinal fluid, as well as the tendency of the compound to crystallize. So the more that dissolves in the low pH environment, the higher the tendency for it to crystallize in the intestinal fluid. So it depends on dose that's put in, as well as the tendency to crystallize. But anything that does not get dissolved in the gastric presumably has an opportunity to dissolve in the simulated intestinal fluid, although it may be slow because the solubility is kind of low there. So quite a few dynamics going on there, but generally it's good to have most of the dose dissolve in the gastric fluid, as long as you can sustain that free drug concentration upon transfer. So hopefully that answers the question.
Okay, thank you. Matt, can cross-link occur in HPMC caps?
No. So a traditional cross-link is either thermally based for gelatin, making it cross-link within itself, which does not happen in an HPMC, or it happens when there's some ingredient like an aldehyde or a peroxide or something like that that would cause the amino acids in the gelatin strands to link between each other. And HPMC being made of a cellulose material instead, it just doesn't have that same chemical feature that would allow or engage cross-linking. So no.
Okay, Matt, can HPMC caps be customized for special applications, such as adding hydrophobic additives to reduce moisture vapor transmission or delay release of the contents?
So for the second part first, for delayed release, a couple different things can happen. You could add some agents to it. We have some experience with adding those gelling systems back. We want to engage in a delayed release type situation. Or I guess you could also coat the capsule easily as well if you wanted to delay it. Otherwise, adding extra things to it, I can't say right offhand. It all depends on the film forming properties of the material. And unless we investigate the addition of some of those in particular, we really won't understand the effect that it has on the thermo-gelation and the ability to form the film polymer. So I would say it's on a case-by-case basis for sure, and be happy to take that up. Just email me or give me a call in the office and let's talk specifics and see if it's something that we've taken a look at in the past or if it's something that we could take a look at in particular.
Okay, thank you. Here's another question for you. What is the importance of the different dissolution media in the in vitro studies when you were talking about gelling agents?
Okay, each of the different dissolution medias with respect to the gelling agents, they're going to be comprised of different ionic activities and often different pHs as well. And depending on the gelling system used, you can see some interaction between pH with the gelling system, and you can certainly see differences in the interaction between the ionic strength of gelling systems as well. So whenever you've got a gelling system and you're concerned about variability from either a pH or an ionic strength, anytime a gelling system is involved, you have to worry about that variability. That's why we really believe in the power of the HPMC Vcaps Plus, the thermo-gelation. By taking out those gelling systems, then that interaction is effectively negated.
Okay. Can you identify some situations where it would be more challenging to formulate with hard gelatin capsules compared to HPMC capsules?
For hard gelatin, the ones I would say the watch it, if you wanted the red, yellow, green, the warning cautionary areas are anything that could potentially contain an aldehyde or a peroxide because gelatin could cross-link. So we want to be wary of that. And we will also want to be wary of any situation that's a water-sensitive molecule may be formulated with gelatin. Because of the amount of water that's critical for gelatin, we want to pay attention to that. Are we using something that is going to be hygroscopic, that could possibly remove water from the gelatin capsule and make it brittle? That's another opportunity. Or is the product eventually going to be stored in some sort of environment that's dry? That would be another, I would say, a cautionary area that we'd want to address. Some of these you can address with packaging, some that you can't. So those would be, I would say, the watch-its from a gelatin perspective, and probably more suitable to work with an HPMC.
Okay, one final question for you so we can wrap up quickly. What products are currently encapsulated with Vcaps Plus?
Okay. Well, there's several, as I mentioned, that are in pharmaceutical development, but there are 4 marketed products at least right now. There's one for major depressive disorder. There's one for ulcerative colitis. There's an oncology agent, and also a generic proton pump inhibitor. Those are all marketed products in the Vcap Plus. So it has been vetted out as a commercially viable product.
Well, thank you very much. Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event, and thanks to both of our speakers. I'd also like to thank our sponsor, Capsugel, for making today's educational webcast possible. This webcast will be available for on-demand viewing through January of 2017. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. Please invite your colleagues to view the on-demand version by forwarding them the announcement. For now, thank you very much for joining us, and see you next time.